Good morning, everyone. My name is Anshita Ratmanshi, Senior Assistant Officer, Abu Banasir University, Lucknow. Today, my topic is on finished product and packaging material, that is granularity of TT process in finished product and packaging. Earlier, we have discussed about granularity of technology transfer process in starting material, which includes active pharmaceutical ingredients as well as excipients. Now, we will discuss about granularity of technology transfer process and finished product and packaging material, which has direct contact with the human. We only intake finished product and packaging material. Packaging material has direct contact with the product which is packed in it, a product which will be administered by the human beings. So this is a very important process, and in this the technology transfer includes very crucial points which we will discuss. In finished product, we can say that the sending unit should provide a detailed characterization of product. What is finished product? As we all know, the result of the outcome of our process is finished product. So it should provide, the sending unit should provide a detailed characterization of or qualitative and quantitative or physical description, method of manufacture, in process control, control methods and specification and all, which all comes under finished product. All the data should be provided by sending unit to receiving unit. Like what is its physical form? It is solid, liquid, and what is the route of administration? In case of solid, it is oral, but in case of like if it is in inhaled doses form, then what is the specific definite form of whether it is in gaseous form or it is in powder form, which is to be inhaled. So each and every description should be clear about finished product. So or we can say of its physical form. Next, in-process control. If I say about in-process controls are the parameters which is being checked during process or formulation process. Like if we talk about tablet, then in-process measures of tablet are its hardness. Time to time, the quality person present there check the hardness, its solubility, its disintegration time. All these process of which are the evaluation processes are being checked between or during the process formulation. Next is solubility profile. If it is for solid, what is the solubility? If it is inhaled and it is transdermal doses form like its patches and all, then what is the solubility profile of that partition coefficient, especially for the determination of transdermal doses form. So, and the next is like development, clinical development, example, information on rationale for the synthesis, what was the exact formula for the synthesis, route and form selection, technology selection, equipments, clinical test, and product composition. Next, scale-up activities, if we have performed any scale-up activities. All these things are being included in technology transfer of finished product, which sending unit should, the data which sending unit should provide to receiving unit. Next is intrinsic dissolution rate, including the method of determination for transdermal doses form. Particle size distribution, including the method of determination for solid, inhaled, and transdermal doses form. Bulk physical properties, including data on bulk and tap density, surface area, and porosity as appropriate for solid and inhaled doses form. Compaction properties for solid doses form. Okay. And also here, like if we talk about scale up of these types of formulation of any of the formulation, whether it is solid, inhaled, transdermal, then in scale up activities, information on scale up activities, process optimization, statistical optimization of critical process parameter, critical quantity attributes, pilot report, and of information on pilot scale development activities, indicating the number and disposition of batches manufactured should be provided by sending unit to the receiving unit. If any changes done, change history and reasons, example change control log indicating any changes to the process of primary packaging or analytical method as part of process optimization and improvement should be provided. Melting point range for semi-solid topical dosage form should be checked and provided that if it in sending unit to receiving unit, like if I'm getting a melting point of any product is 128 degrees Celsius, and if in receiving unit also the melting point for the prepared formulation should be between the range provided by the sending unit. So all these types of range should be provided. pH range for parenteral, semi-solid, topical, liquid and transdermal dosage form should be provided. Ionic strength for parenteral dosage form, specific density, gravity for parenteral, semi-solid, topical, liquid and transdermal dosage form, viscosity and viscoelasticity for parenteral, semi-solid, topical, liquid and transdermal dosage form, osmolarity for parenteral dosage form, water content and determination of hygroscopicity, including water activity data and special handling requirements for solid and inhaled dosage form should be provided by sending unit to the receiving unit. Now next is packaging material. Information on packaging to be transferred from the sending unit to receiving unit includes specification for a suitable container, closure system, as well as any relevant additional information on design, packing, processing or labeling requirements needed for qualification of packaging components at the receiving unit. In this, the transfer of packaging operations should follow same procedure patterns as those for production transfer. In information of packaging to be transferred from sending unit to receiving unit, especially include, as I have told, containers and closure system. What are the specification? Color, strength, and these are the basic color, strength, size, height. Diameter, everything of packaging system, thickness, hardness, if it's of foil and all, then all these types of specifications should be provided from sending unit to receiving unit. That to state that in sending unit, this technology was including this type of specification of packaging materials, so it should be considered in receiving unit also. The next is quality control testing of packaging component specification should be provided for drawing artwork material. Drawing artwork material is if I'm talking about containers and cartons and packaging or labels, etc., then which type of artwork is done. It is similar to sending unit or not. If sending unit includes light yellow or brown color of label, then in receiving unit also the final product should include that specific yellow color which was provided by sending unit. The color should not be lighter or darker. Okay, and if it has included only two colors, then in the receiving unit also the label should include that only two specific colors which was provided by sending unit. Based on the information provided by receiving unit, it should perform a suitability study for initial qualification of packaging components. Okay, and next, packaging is, as we all know, that packaging is considered suitable if it provides adequate protection, preventing degradation of medicine due to environmental influence, safety, absence of undesirable substances released into the product, compatibility, absence of interaction possibly affecting medicine quality and performance, functionality in terms of drug deliveries, etc. Okay, so these all are basic information which is provided by sending unit to the unit when technology is transferred. Thank you for your cooperation.